Hello children, so today we'll be doing experiments on photosynthesis. Here we have 4 experiments that we have to prove, and please remember that it is the continuation of the same chapter, that is photosynthesis. So the 4 experiments that we have to prove, the first one is the experiment to prove that chlorophyll is necessary for photosynthesis. Second one, sunlight is necessary for photosynthesis. Third one, carbon dioxide is necessary for photosynthesis. And the fourth and the last experiment is oxygen is liberated or released during photosynthesis. So now we will be doing these 4 different types of experiments on photosynthesis one by one. But one very important thing that you all need to remember is that, and to keep it in mind, out of these 4 types of experiments on photosynthesis, the first 3 experiments, that is chlorophyll is necessary for photosynthesis, sunlight is necessary for photosynthesis, and carbon dioxide is necessary for photosynthesis. Here we have 2 important steps or processes which is common to all these 3 types of experiments. So please remember this one. But the fourth experiment that we have, that is oxygen is released during photosynthesis, here we have to perform different experimental setup than the other 3. So please keep this in mind. So first I will explain the 2 important steps which is common to all the first 3 experiments. That is, the first one is destarching, and this is the first step that we have to do before doing the experiments. And the second common step for all the 3 experiments again is iodine or starch test, and we have to do this or perform this process or the step at the end of the experiment before we get the observation or before we see the observation. So please remember the first and the second steps in every experiment, and these 2 steps are very important for your board exams because they set many questions from here, or they give in paragraphs to fill in the blanks, or they ask you to write down all the steps in a sequence. So once again, let me remind you that whenever we are performing experiments on chlorophyll is necessary for photosynthesis, sunlight is necessary for photosynthesis, and carbon dioxide is necessary for photosynthesis, we must have to follow these 2 steps or processes. Now please keep this in mind. So now let me explain these 2 important steps, and please listen very carefully. The first step is destarching, which means removal of starch. And what is this starch? Starch, it is the stored food in the plant body. So destarching means removal of starch from the plant body. So the first process or the step that we have to follow in this destarching is we have to remove the starch from the plant before performing an experiment. Why? This is the first question that they usually ask. The answer will be we have to destarch the plant before performing an experiment to prove that all the starch is removed, and whatever starch is formed now while performing an experiment for starch test, all the starch is formed during the experiment. And secondly, how do we destarch the plant? This is again another question. We destarch the plant by placing the plant in the dark for 24 to 48 hours or 1 to 2 days. And what happens when the plant is kept in the dark? Again, there is another question. So when the plant is kept in the dark, this plant cannot prepare the food by the process photosynthesis, or photosynthesis cannot take place. Why? Because of the absence of sunlight and water, since it was being kept in the dark. Again, what happens then? The answer will be, so whatever starch was stored or was being present in the leaves of the plant will be used up by the plant to survive, whereas the extra starch will be removed to the different storage organ from the leaves. Therefore, there won't be any starch in the leaves of the plant. So please remember, so no starch will be there in the plant when it is being kept in the dark. Now thirdly, now what we have to do is, now we have to keep the whole plant outside in the sunlight again. Why? For restarching. That means photosynthesis will take place and then the starch will be formed again. Now there is one important question from here. Please remember this. Please take a screenshot and copy it in your exercise copy. The question that we have here is, why do we destarch the plant? We destarch the plant to prove that there is no starch in the leaves of the plant before the experiment was performed, and all the starch that is formed now while performing the experiment for starch test is produced during the experiment. So please remember to whatever I said just now. So this is all about destarching and the questions relating to destarching. So please note these points in your biology exercise copy, and I have often told you that it is very important for your board exams. Now we move on to the starch test or iodine test, which is the second step. So we all know that whenever we pour iodine solution on any plant parts, or say a cut potato, etc., it will turn blue black in color. That is very simple and that is seen also, but we need to follow certain steps. So please listen very carefully and remember these 5 steps that we have to follow in a sequence while doing the starch test, and not haphazardly. Okay, during the exams they might ask you to write down the 5 steps in a sequence, or they will give you these 5 steps in a scrambled term and you have to unscramble them in a logical sequence. This is how they ask. So now I will be discussing regarding the 5 steps that we have to follow while doing the starch test. And the first step is we have to boil the leaf in water. Why? To kill the cells. And one thing, please remember the reasons also why we are doing this. Second step is, now we have to boil this leaf in a methylated spirit or an alcohol. Why? The reason will be to remove the chlorophyll. And how do we know that the chlorophyll is being removed from the leaf? The leaf will turn pale white, and since we have dipped this leaf in an alcohol or in a methylated spirit, the leaf will now become brittle and hard. You know what do you mean by brittle? It can break into pieces if you hold it very firmly. Now this brittle leaf is now dipped in hot water. Why? To make the leaf soft. Now the last step is, this leaf must be spread over a petri dish or a watch glass or any kind of saucer or a plate where we have to pour iodine solution. So the part containing the starch, or the portion of the leaf which was green in color, turns blue black, which shows that there is the presence of starch, while the portion of the leaf where there was no starch, or the portion of the leaf which was not green in color turns brown in color. So this will prove the presence of starch in a leaf by the change of color to blue black. So please remember, the blue black color will indicate the presence of starch in a leaf. Now we will do the experiments in a sequence. That is, we have 4 experiments. So we will start with the first one, that is experiment to show that chlorophyll is necessary for photosynthesis. Here in this experiment we have to take a plant with variegated leaves. So please remember this important point here. So what kind of plant do we have to take? We have to take a plant with variegated leaves while performing an experiment to prove that chlorophyll is necessary. And what do you mean by variegated leaves? It means the some portion of the leaf will be green while some portion will be non-green. And please remember the name of the plant with such variegated leaves. They are geranium. Here I have shown you one diagram, and I think you know this plant and you all have seen it. It is an ornamental plant. Next example will be coleus croton. So please remember the names of these plants with variegated leaves for 1 mark during the exams, and please mark it in your textbook also. Now we will be doing how to perform this experiment. First is, the whole plant is kept in the dark for 24 to 48 hours. Why? To destarch the leaves. And how to destarch, why are we destarching it, that I have already discussed you earlier. After destarching the plant, the whole plant is then placed in the sunlight for few hours. Now why? That also I have already discussed, that is to restarch the plant. Then any one of the variegated leaf is plucked and an outline diagram of this plucked leaf is drawn on a piece of paper showing the green and non-green portion of that leaf. Next is, this variegated leaf is now tested for starch by following the steps as I have mentioned few minutes earlier. So please remember the names in a sequence. They will not ask you to write while performing the experiment. After that, what do you see or what observation do you get? Now please remember this one. Only the green portion of the leaf turns blue-black, showing the presence of starch, whereas the non-green portion turns brown in color, which shows that there is no starch formed. So the green portion which shows that there is the presence of starch means to say that the process of photosynthesis has taken place during the experiment. So what conclusion we have, or what can we prove, or what does the experiment prove or show? It shows that, or it proves that chlorophyll is necessary for photosynthesis. Now next point that you have to remember is, please do not get mixed up with observation and conclusion or proof. Observation will be change of color of the green portion of the leaf to blue black, whereas the non-green portion of the leaf to brown. This is the observation that you have to write it if they ask you. And the next will be conclusion, or what does it prove? So the answer will be chlorophyll is necessary for photosynthesis. So from this chlorophyll is necessary, please remember the name of the plants with variegated leaves. So now next we will be doing the next experiment, that is sunlight is necessary. Here also the experimental process will be the same in the beginning and at the end. So the first thing that you have to do is, the plant is kept in the dark for 24 to 48 hours or 1 to 2 days. Why? To destarch the leaves of the plant. Now, here please remember that we have to do something little extra than the previous experiment. That is, we have to take a complete black paper or a black paper with a cut designed on it as I have shown it here. Any design you can make, but we have to cut that design. Okay. Next is, after destarching the leaf, we have to take the plant outside in the sunlight. But before keeping the plant outside, we have to place this black paper with a cut design on it on any one of the leaf of that plant with the help of a paper clip. See the diagram here. Then the plant has to be kept in the sunlight for a few hours. Why? To restart the leaves by the process of photosynthesis. So the question that they usually ask is, why do we have to keep the plant in the sunlight after destarching? The answer will be to restart the leaves by the process of photosynthesis. Next is, the leaf is then tested for starch by removing the paper. As before, we have to follow the 5 steps one by one, and at the end we have to pour iodine solution over the leaf. What observation do we see? The observation will be, please remember this one, the portion of the leaf that was covered by the black paper will turn brown. That means no starch is present there. Why? Because photosynthesis has not taken place due to the absence of sunlight, whereas the portion of the leaf that was exposed to sunlight will turn blue black. Why? Because the photosynthesis has taken place when exposed to sunlight. So what will be the conclusion? What will this experiment prove? It will conclude or prove that sunlight is necessary for photosynthesis. So today I will be doing only these 2 experiments, and then the other 2 important steps that we have to follow before performing the experiments. So please go through this very carefully, and at the same time please go through your textbooks also after going through the video. In case you have any confusion or any doubts, then you may ask. Okay, this much for today. Thank you, class.